So now that we know what the blockbuster drug is, let's talk about how we're going to actually deliver this in private practice all across the country and all across the world. The Personalized Lifestyle Medicine Institute was started by Dr. Jeffrey Bland a few years ago, and their main conference, the Thought Leaders Consortium, has really been providing a 30,000-foot view of the future of medicine. Now what we're going to take a look at in this conference is something very different. How do you practically apply lifestyle medicine in private practice? And we're very excited to bring the keynote for this session from the founder, Dr. Jeffrey Bland himself, as he goes through the basis for a future of medicine built on lifestyle. It was an incredible 45 minutes. We got some of the best parts here. Enjoy.
We're involved in a non-placebo controlled great global experiment in genetic resiliency to a rapidly changing environment. And we're asking the question now, on a human level, cultural, world, global level, how resilient is the human genome to an unbelievably rapidly changing environment? And the news is the yellow canaries are telling not completely resilient, right? Because the yellow canaries are the first individuals with a genotype that is responsive to those changes. It's telling us that it's under alarm. And the conditions that we see prevalent in the world today are disorders that are associated with alarmed genes. They're going into defensive battle against a hostile environment. So when I hear people talk about that person has a disease gene, like for rheumatoid arthritis or systemic lupus erythematosus, an autoimmune disease, I say, no, they don't have a gene for that disease. They have a genetic response to an environment that maybe in a previous era and some distant ancestor gave them selective advantage against an infection that might have killed everybody else but not them. But now in this environment, it becomes a disadvantage, because now you get the collateral damage to the individual from their genes responding as they were supposed to have responded some many generations ago. So I think the term disease almost is a little bit of a, it has an element of discrimination, I think, associated with it, that makes a person feel unworthy or less than whole. But maybe they're actually super whole. It's just the environment they're in is not matched to their genetic uniqueness. Okay, so what is then personalized lifestyle medicine? And Dr. Minnick and I wrote this article a number of years ago in 2013, in which we tried to differentiate lifestyle medicine, which is, by the way, very valuable, to personalized lifestyle medicine, because we feel that the health of the world is really a result of the health of each individual, one at a time. And so if you want to get the world to be healthy, you need a bunch of healthy individuals. And each individual is slightly different one from another in how they respond to the same signals. So the more we can understand what signals turn on the right genes in those people, the health promoting characteristics, the more successful we'll be in creating a globally healthy community. Now that does not mean that public health is not valuable. It's extraordinarily valuable, but you probably know what's going on in public health now is its trajectory is towards what's called personalized public health. There's an intersection then between this two things that were previously separate. One over here was individual patient treatment, the over here was public health, and they never met. Now we're seeing, because of this new body of information that's coming about, that personalized precision public health is starting to emerge. So this ecosystem for personalized lifestyle medicine is an ecosystem with many different components. And I'm not going to go through this diagram in length, but people that were previously not even considered part of the healthcare community. And do you find it interesting that some of the major healthcare innovators now are Google and Apple and Microsoft and Cisco? I mean, you say, really? I mean, these aren't even things in the healthcare community, but they are, because of the information content that is necessary to be digested to produce an understanding of this complex interaction of genes and environment. These companies have some leadership in how that information can be aggregated using machine learning or complex informatics. So there's many different parts of this community that we're starting to learn. These are our friends, right? And starting to converge to a new model of what healthcare would be, including biometrics, including exercise physiology, including, the whole area of behavioral psychology. I mean, I could go on and on and on, but there are many layers on this system that require collaboration. So we see then personalized lifestyle medicine as being part of this schematic view of health and disease that most of us grew up with in this room, in which we recognize that health or wellness can be on a very broad scale, from everything from optimal health on one end to the complete absence of health on the other, which we call death, right? And you can reside on various places within that continuum as an individual based upon how your genes are matched with your environment. And you may be somewhere before you're diseased, so you're just kind of chronically unwell. But generally, if your genes and environment are mismatched, the trajectory goes towards increasing severity, duration, and frequency of symptoms, which will get you eventually to a name of some ICD-10, right? Eventually. So this construct is involved with, let's early on understand the individual and help guide them by collaboration to find the right environment for their genes so that they don't move over that threshold when they are moving through their life process, which we call disease. And so this little cartoon kind of diagrams or symbolizes everything I'm talking about, that our information content, our legacy and our book of life is being impacted by signals that come from all sorts of things that turn on different messages within our genome. Some of those messages are alarm messages, other messages are bliss messages, and the bottom line is that most of us know where we are on that interface. If we really tune into our body, we can kind of tap into where we are on that sequence of events of how our genes are responding to our environment. Now let me go off the reservation here for a second and raise a question. Do our genes necessarily in themselves, are they hardwired to produce a specific phenotype? And that's an interesting question, right? So do we have like depressive genes, so you're just going to be depressed because you've got those genes? Or are you going to be an extrovert because you have extrovert genes? Or an introvert will never be an extrovert. I mean, these kind of archetypal Myers-Briggs personality characteristics. So again, I'm going to go back to dogs. Great teacher. I've often said that if you could take the genes out of my golden retriever, our golden retriever, and insert them in humans, we'd have no wars. And everyone would love one another. And so what is it about dogs? Because dogs are very close genealogically to wolves, right? And in fact, if you do genome screens, a dog, their genome is as close to a wolf's genome, actually even closer, than we are to chimpanzees. And as you probably know, we have 97.6% homology with chimpanzee genomes. So you start to say, well, hold up, my dog really doesn't behave like a wolf, so what's going on? Well, it's been found that there are two genes that are epigenetically modified by methylation in the wolf genome to produce the dog docility, right? A dog that's loving. Epigenetic effect of environmental, lifestyle, and genetic traits, right? This is another part of our story. So we have this big tuning knob called our genome. Then we have a fine tuning knob called our epigenome, which are marks that we put on our genome from experience that we have that then regulates how our genome is going to be expressed. Am I making sense here? So we really can't change our genes, but we can change aspects of our genomic marking that we call epigenomic that then patterns how our genes are going to express themselves. So going back to our dogs, domestic dogs, the concerted impact of domestication is on these transposon insertions by epigenetic modulation of the wolf genome to produce the wonderful personality of our domestic dogs, right? So what happens if we took humans that were more like wolves and made them like domestic dogs by the right environment? So I think that you can think of all sorts of metaphors here, and you actually, like in movie scripts, like The Wolf on Wall Street and all sorts of things, you know, about, so what kind of messages do we send to our genes to create our behavior? Now let me give another example. This is one that's very topical. The football season is upon us. So we all know about the traumatic brain issues now that are undoubtedly going to change American football significantly over the next years to come, probably through litigation. And so that will change the way football is played, I'm sure. And so you might say, well, what is this all about? Well, it turns out that if you look at the data, and these are the JAMA papers that you probably saw about the clinical pathological evaluation of chronic traumatic encephalopathy, CTE, in American football players. This is high school, college, and pro players that you've heard so much about, that the brains were shown on that previous slide, that individuals had died and willing to have their brains pathophysiologically evaluated. And what they're saying is that the signals that these brains get from these concussive injuries are such that they set in motion a degenerative condition that can resemble Alzheimer's, right? Or other kinds of situations involved with dementia. Now let me think about that just for a second in the context of what I've just talked about. How does that work? So you have a mechanical injury, right? A mechanical injury is that this organ is slammed against the skull through this impact, and it suffers some kind of a bruise. Now, we think of a bruise on our arm healing and going away, and then our arm is fine. But this particular injury has a replicative sequence over time that doesn't get better. In fact, it gets worse, and these people don't get dementia immediately. Their brains become more fungiform over the course of 20 or 30 years, post even their career as football players. Now, what is going on there? Think about that for a moment. The only way that that can happen is if you've epigenetically changed gene expression. So we have a mechanical signal called concussive injury being translated into a progressive gene expression change of alarm in the brain, in which the collateral damage is the neuron itself that leads ultimately to neuronal depletion and dementia. Now that's a very different way of thinking about these things, just in a context, this lens of thinking about genes and environment, because now we're talking about progressive, something that goes on in the absence of the stimulus. It continues on to progress because you've reset gene expression patterns in a new homeostasis, which is the homeostasis of degeneration. We often think of homeostasis as a term that refers to proper blood sugar management, blood gases, blood electrolytes, around those normal values. But homeostasis could be the homeostasis of alarm and inflammation and hyperglycemia. And so how do you break that steady state, the homeostasis of dis-ease? You've got to reset the genetic programming that's epigenetically been modulated through that event. Okay, so that's why this, I believe, has a personal characteristic, because we know that if you happen to be carrying an ApoE4 genotype and you have a concussive injury, because those ApoE4s make you more inflammation prone. There's a whole story as to why ApoE4. Do you realize the human genotype probably early on was mostly all ApoE4? And the ApoE2 and 3 are more recent developments, because in our early hominid evolution, it was very desirable to have high inflammatory response to infection. It was the greatest threat to, and that ApoE4 is really good for responding to infection. Unfortunately, it's also responds to other triggers that produce inflammation, like high saturated fat diets, which we didn't probably worry about in early hominid history, but now we do worry about, right? There's a new signal impinging upon that gene that creates now not a beneficial outcome, but a disadvantageous outcome. So if you have an ApoE4 and you have concussive injuries, your risk to CTEs go up significantly, right? So here is where it becomes personalized. Not every football player gets the same degree of CTEs, like not every smoker ends up with lung cancer. Okay, so this genetic uniqueness is very, very important. And as Eric Topol points out, he's the author of several books, but one is The Patient Will See You Now, which turns this whole thing around, saying medicine is going to be consumer-driven as we move forward. And these omics tools, which are genomics, epigenomics, transcriptomics, proteomics, metabolomics, and metagenomics on our microbiome, all will be integrated together in this broad thumbprint, fingerprint of the individual as we move forward in healthcare, so that we know the individual at a much deeper level than we have in the past, which we've just kind of speculated based on the way they present signs and symptoms. And that will be tied together with the biomarkers to give a new mosaic of understanding of the individual. So it's the start of making wellness precise. And this is, as you probably know, I've been working over the last few years with Lee Hood and his group at the Institute for Systems Biology in this Pioneer 100 project, where we've each of us, 105 participants, have had several, I think in my case, a little over 3 million data points collected over the last 3 years on this routine and kind of analysis we're doing every quarter. And then that goes into a big informatics system to evaluate, trends. And the first paper of the results of the Pioneer 100 project was just published this summer in Nature Biotechnology under the title Wellness Study of 108 individuals, using personal dense dynamic data clouds. That's a Lee Hood term, that all this information goes into the cloud, where it's then stored so that then informatic analysis can occur to give us guidance about our own unique states of need to match our genes with our environment. So there are new methods of proof in the age of complex lifestyle diseases. We're starting to go away from the randomized clinical trial to looking at things that come from the citizen scientist. That's an interesting term, isn't it? The citizen scientist who's on the internet and wants to be part of studying themselves. So they are willing to sign on to their data, like 23andMe. People say, yeah, you can use my data agnostically in a data set for analysis. So a citizen scientist is changing research methodologies, and it's changing the way we think about how to aggregate data to produce complex data sets that give multivariate abilities to evaluate rather than univariate, because the way that the RCT worked is often univariate analysis, one variable against one outcome. And it's really great for a drug against a disease, but it's not so good for lifestyle complexity where there's so many things going on simultaneously. So new methods are becoming now standards. And this New England Journal of Medicine paper last June really says, hey, we have to go beyond the RCT if we're going to really answer these complex questions about how people really get sick in environments that are not exactly matched to their genes. Now, how important are the things that relate to diet and environment and outcome in a disease? Well, we're seeing a huge number of new papers come out in top-level journals that are showing this. This is a paper that just was published in the JAMA recently, showing that poor diets contribute greater than 50% of the risk to heart disease, stroke, and type 2 diabetes. So these are not small modifiers, these are large modifiers. And we now recognize that red meat, sodium, and processed foods are major associations with premature death. This was, again, talking about attributing death to diet and the fact that we need to use statistical precision. But when we do so, we actually can see that these are really important contributors to the way people's genes translate into their health or disease over lifestyles. So diet is a large driver for altered physiological function. This is a New England Journal of Medicine paper saying a 20% improvement in diet scores was significantly associated with a reduction in total mortality between 8 and 17%. And those that were the lowest quintile of diet quality, they could have up to a 20% improvement in their life expectancy by changing their diet. Now, there is no drug that you can find on the market that can deliver that kind of benefit. So here's another paper that was just published in the Journal of American Medical Association, looking at intensive lifestyle intervention on glycemic control in patients with type 2 diabetes using hemoglobin A1C as the marker. And so the yellow line you see there in the graph is standard of care, which is 1 or 2 anti-diabetic drugs. And the dark lines are lifestyle intervention over the course of a year. And what they show clearly is it's as good at lifestyle intervention is as good or better than standard of care with lowered incidence of adverse side effects. So diet and exercise, lifestyle speak to our genes. And in fact, this question that was raised in what is arguably considered the top journal in cell biology called Cell, the article was entitled, "Will Diet and Exercise Save Us All?" ? And the answer to that, if you read the article, says it's likely it will if we can just implement this. Now these are the fundamental highbrow scientists, right, doing the basic science research on cellular biology and physiology, who are coming to the conclusion from all this data that yes, this is how powerful these signals are. So will it improve patient outcome? And the answer is yes. Personalized nutrition by prediction of glycemic response. This is work that was done in Israel you're probably familiar with, showing that prediction based upon these personalization, genomic and phenomic information take on patient was accurate and superior to common dietary advice, and they could do a much better job in guiding people away from type 2 diabetes by using personalized intervention than by using community-based population information. And what kind of diet do we think about? Well, as you probably know, the topic of the day right now are these fasting mimicking diets, or FMDs. Very, very powerful information now at the cell-based levels in both animals and humans, showing that if you can keep from eating for at least 12 hours each day, that you're doing some very good things to your beta cells and regenerating their insulinogenic capability. If you can move yourself into this kind of what probably was the way that people had to eat, or did eat for much of our human history, which was not available calories at 3 squares every day with snacking in between, but rather this fasting intermittent intervals. And when we're talking about fasting, we're not talking about like cold turkey fasting. Here we're talking about, and we're not even talking about what's called calorie restriction. We're talking about the rhythm of life, a temporal use of eating in such a way that you're not eating with throughout the whole day period. You have at least a 12-hour rest from the last time you've eaten until the next time you eat. So say 6 to 6 or 8 to 8 or whatever it might be. And this fasting mimicking diet has been shown in human intervention trial. This is a paper I'm showing you there, Science Translation of Medicine, to actually improve every metabolic parameter in humans within 3 months that is associated with diabetes, stroke, and heart disease. So it alters the way genes are expressed by just circadian rhythmically controlling calorie consumption. So that is regulating network biology and regenerating beta cell function with diet. And again, I cite this article in JAMA, just appeared in April issue, talking about can a diet that mimics fasting turn back the clock? And the answer is yes, apparently, it can. And in fact, in this article in Cell, it actually shows in human and animal models that it regenerates beta cell function in people that have lost insulin secretory ability. Now, this is powerful stuff, right? It would be like getting a beta cell infusion. So newsflash, bioactive ingredients are found in food, and therefore one asks the question, where does food stop and drugs begin? Because we're modulating function in individuals as powerfully as drugs would do, but we're doing it by normalizing network biology, not just blocking one metabolic step in our function. So the influence of bioactive ingredients on intestinal immune function are a good example, and now we start building in an understanding of the microbiome and its relationship to the immune system that you're going to hear much more about over the next couple of days. This again is, I'm trying to show you the level of article journals that these are appearing in. New England Journal, a whole review on the microbiome and its relationship as a gateway to the immune system, and the metagenome of a diet and in the relationship to an individual's diet can, in fact, look at the title of the one paper, Veggies and Intact Grains a Day Keep the Pathogens Away, right? So we're really talking about this complex matrix, this symphonic orchestration of how our lifestyle principles regulate all sorts of functions, not just one step at a time, but like conductors of the orchestra of our physiology. So what makes the microbiome so important? Well, it's a bioreactor producing many bioactive compounds, right? Not just short-chain fatty acids like acetate, propionate, and butyrate, but it also produces all sorts of other interesting compounds that regulate through receptors on our immune system that are localized in the gastrointestinal-associated lymphoid tissue, the gut immune system, to regulate systemic signals to all the genes of our body, in the neurons, in the liver, in the beta cells, and all of these are getting a different orchestrated effect. And in fact, one of our colleagues, Patrice Connie, that we've collaborated with at Catholic University in Belgium, this paper that he and his colleagues just recently published showed that one of the interesting organisms in the gut microbiome, called Akkermansia muciniphila, that it has in its cell wall a protein that if you were to sterilize, in other words, you kill the bacterium, you take that cell wall extract and you give it to animals, treats diabetes, right? So these very unique compositions of the bugs that live in our gut are communicating in multiple ways with our receptor systems to produce function, basically, systemic function. And we also know they're involved with sending on secondary chemicals that are involved with hepatic detoxification. So if you consume drugs, it's not just the drugs itself, but it's the metabolic byproducts of the drugs that come from your own microbiome that lead to different responses to that drugs. Some are adverse side effects because that person, by their bugs in their gut, produces secondary chemicals that are not so good for the individual. These are things we never even thought about, by the way. We used to just say, oh, that person has an unexpected adverse side effect to the drug. Well, it's really probably not unexpected if you ask the right question. It's probably to be expected, and it's reproducible based on the way their body metabolizes and responds to that medication. And then we know that the gut, through this complex signaling mechanism, ties itself to the brain, to the enteric nervous system. And as Michael Gershon told us many years ago, the gut is the second brain. And because the gut produces things like the enteroendocrine hormones, called the incretins, like glucagon-like peptide 1 and ghrelin. And I mean, you have all these various interesting things that are being produced that are neuroactive from the gut that regulate gene expression and function. You produce neurotransmitters. You probably know the biggest source of serotonin in the body comes from the gut enteroendocrine cells. And lipopolysaccharides and the gut bacteria, these cell wall debris will activate the pattern recognition receptors, the so-called toll-like receptors. So the point I'm really leading you to as we bring this to close is that there is a very, very profound interrelationship between our specific environment, our unique environment, and how our unique genome, book of life, responds to give rise to our health and disease patterns. And that's why we advocate moving to a personalized focus about how we're going to deliver lifestyle healthcare. And I was very pleased to see just this last week in JAMA, Alessio Fasano, one of our great leaders of the field at Harvard, doing the work on gluten enteropathy and celiac disease, believe it or not, was able to get through the referees of JAMA this paper that talks about celiac disease and non-celiac gluten sensitivity. Now I want you to remember, it was just 5 years ago that JAMA had a paper saying there was no such thing as non-celiac gluten sensitivity. You either have celiac disease or you don't. There is nothing in between. Now, this paper clearly defines the frequency of response, adverse response that people's immune system have to gluten-containing grains is much greater than one would have expected on the basis of just celiac antibodies alone. And this non-celiac gluten sensitivity is a real phenomenon. What I'm saying is, this is the opening of a new age to recognize the importance of individualizing lifestyle to produce healthy outcomes, and it will transform medicine. And I'm privileged to be still part of this extraordinary process that will deliver to people benefits, and as Linus Pauling said, our objective is to reduce human suffering. And this particular approach, this philosophy, allows us to move to the most common conditions of concern globally, which are non-infectious lifestyle-related diseases. Thank you very much.